Oh, there we go. That's good.
No, it's not on the.
Can you advance that? All right.
Okay, okay.
Don't wait for a second.
Wait, does the clicker work? Yeah, the
All
clicker
right.
works. All right.
The clicker works. Right. So Julie did her undergraduate degree in marine science at the University of Miami for 2016. She took some time off and she arrived in our lab and in the Ecology and Evolution program in 2018. And here she is today after a few years of work. I've gotten to know Julia really well. And what I can tell you is that I know she really relies on her family and friends. So thank you all for coming. She's always spoken very highly, and I've met a bunch of you and I can see why. So that's great. And all the folks on Zoom who are listening in, and all the research colleagues at Rutgers and elsewhere. So it's been a real pleasure to hang out with Julia for the last few years, and I'm sure we'll have lots of fun years coming up. I just want to give you a little bit of introduction into Julia's time here, because she's not a nose to grindstone only grad student. She's actually done some really cool stuff with her life, and just a few highlights from her graduate career. And this is just sort of scratching the surface. First of all, she's an activist. Yes, she's an activist for social causes, but she's very importantly an activist for what we in science call horizontal gene transfer. That is the movement of DNA across species and not through the parental and daughter lineages. This may sound odd to you, but it's a big piece of what Julia's research was. And the paper she wrote in Trends in Genetics has over 100 citations already. And it's kind of marks the break between the old time when we didn't believe this process was very common in eukaryotes to now when we take it more seriously. But that's a really cool thing. So she's an activist for that. She's also an early earth scientist. She was funded by the NASA Finesse program, and you'll hear from her talk that she's done a lot of work on sort of environments that are proxies, or we believe were similar to the early earth, and they provide models. So she's done a lot of work with that, and Lynn Rothschild has played a role in that as well. And so this is one of her interests. She's an award-winning systematist. Who would have thunk it? She wrote a paper on using a novel method called Cambridge she'll talk about that ended up winning the best paper of the year by making a significant contribution to systematics research, which is, I thought, very, very impressive. And she's a thought leader. She was a member of a very august panel in Chicago at the Field Museum, which unfortunately included me, in which we discussed the future of symbiosis research. Nepotism, right? She's a Nepo thought leader, and there we discussed, how can symbiosis research help the conservation programs that are needed due to climate change. And she's an amazing educator and outreach artist extraordinaire. She has this podcast called Tiny Living Beings, and she's been in films with Rutgers, so she's done a lot of stuff and she's helped design a course for microbiology program that are all trying to bring the beauty of algae and produce to people. So that's really important that the next generation learn some passion for these cool organisms, just like Julia has. And of course, she's the couch and field microscopist. She's done an amazing sort of Instagram site, not beyond the number of people enjoy it, but just the fact that she's educating people about these tiny things we don't see with our eyes. We need a microscope, but they're critical for how our planet functions. She is also an obscure algae specialist. She wrote this really cool opinion paper about how we need to focus on the weird little things we don't know much about. And that's actually another really important contribution, to stop thinking about model systems and think about organisms that are less known, but can actually have really important insights into the biology of our planet. She's an entrepreneur. She's got a bunch of side hustles going on to talk about, but you can buy some of these things for a couch microscopy website, which I think
Thank you.
We're partners now. And finally, she's a famed impressionist. So I was made coerced not to unearth the Bernie Sanders audio I made during the trip to North Carolina last year, which is hilarious. So I chose a much more sane route, playing Carl Sagan in the Sagan-esque turtleneck, showing the book last Halloween. So Julia, she'll talk a lot about her science, and that's cool, but I just wanted you all to know that she's done a lot of really great things with her time at Rutgers, really sort of important things for outreach, for helping people understand the beauty of the natural world. And I think that's a great contribution from anybody, especially a grad student. So with that, I'll stop and I'll leave it to Julia to take over.
I think everybody can see. They can see the notes. I have them here if you want to. Thank you. That was really nice. Are we good? Okay. All right. Thank you, Debashish. That was very kind. I'm also a very nervous public speaker, so we're going to get through this together. Thank you everyone for coming to my public dissertation defense. It's called DNA Transfer as a Driver of Eukaryote Genome Evolution. Oh no, the clicker is not working. Let me try to. Okay, I'll work with this. Hold on. Let me see if this works. Nope. Oh, yes. Okay, great. So today I am going to present my 4 dissertation chapters that are individual projects I completed over the last 5.5 years in the Bhattacharya lab. But first, I'm going to give a little background on the major concepts and history that are important to understand before I get into detail. So horizontal genetic transfer, or HGT as I'll be calling it for the is the acquisition of foreign DNA by a genome within a generation. Additions of new DNA, which could encode genes or not, lead to new genetic variation within populations. Some gene additions may confer an instant functional advantage. So if a stressor was to come along that could only be survived with the help of that HGT encoded function, the old population may die out and only those with the HGT are going to survive and reproduce. Oh, whoops. It's long been accepted that prokaryotes, so bacteria and archaea, exhibit high amounts of HGT. And on the left here, we have a bacterium that's dividing and passing a copy of its genome onto its offspring. And this is the canonical vertical way that genetic information is transferred. In contrast, on the right, I'm going to show a few examples of how that same bacterium could receive new DNA into its genome horizontally, or, oops, the animations didn't work, but okay. Horizontally or within the generation. Oh, I think this is the old version of the PowerPoint, but I think it's okay. All right. These include conjugation, which is essentially bacterial sex, transduction, or when a virus injects its genetic material into the cell, which could then integrate with the genome, and then transformation, where a cell with an ability to take in foreign DNA may pick it up from the external environment. In eukaryotes, the process is very similar, except that a foreign piece of DNA must overcome some additional hurdles to become a fixture within a new cell. So first, eukaryotes have a walled-off nucleus, so this barrier must be passed to reach the genome. And second, if we're considering multicellular eukaryotes, HGT can occur in any cell in the body, but it's only going to be passed down if it enters the germline prior to reproduction. So for these reasons and others that I don't have time to discuss today, HGT in eukaryotes was long dismissed as rare and only associated with specific events in eukaryote history, like organogenesis. In fact, the year I started my PhD, there was a publication titled Eukaryote HGT is Lamarckian. Speaking of Lamarck, he posited that organisms acquired physical traits within their lifetimes that could somehow be inherited by the next generation. However, Darwin successfully challenged this idea by introducing the descent with modification paradigm that invokes natural selection as the gradual mechanism by which species evolve over time. By the 1920s, scientists, including Frederick Griffith, had observed some rapid, not gradual adaptations in disease-causing bacteria that they were studying. And we now know this as transformation, and that's an example of HGT. In the 1960s, Lynn Margulis, my favorite scientist, established the theory of endosymbiosis, and that explains how eukaryotes and algae evolved by chimeric associations between bacterial endosymbionts and archaeal or eukaryote host cells. So in other words, she showed some proof of some major examples of eukaryotic HGT. Many neo-Darwinists had problems with Margulis for a bunch of reasons, including that they also just didn't like anything that challenged Darwin's ideas of gradual change. But By the 1990s, we had Carl Woese, who used DNA sequences to reconstruct the phylogenetic history of the 3 domains of life. And that kind of analysis, combined with the ongoing genomics revolution, has led to mountains of examples of eukaryote HGTs that have been validated. So the few remaining dissenters will argue that HGT into eukaryotic genomes is Lamarckian, but that's completely untrue. HGT occurs on the DNA and not phenotypic level, and it's subject to the same Darwinian forces of evolution as any mutational change would be. So we now accept this new paradigm that the tree of life is a web of life, but we're also just at the beginning of understanding HGT as a fundamental driver of evolution in eukaryotes. And so this is what I begin to address through my dissertation work. In my first chapter, I review the adaptive role of HGT in eukaryotes with different lifestyle strategies. In chapter 2, I validate the function and integration patterns of ancient gene transfers in some red algae. In chapter 3, I apply an alignment-free framework to place microbes from environmental sequencing projects into the tree of life. And in the final chapter, I apply a similar framework to some of our genomic data from Yellowstone National Park to show patterns of DNA transfer across an entire ecosystem. So chapter 1, I just took a sip of water. So chapter 1 is called Horizontal Gene Transfer in Eukaryotes, Not If, But How Much. And this was a review paper that was published in Trends in Genetics all the way back in 2020. I want to see if I can I use like a laser on the screen? Whatever, all right. We're gonna go without pointing to anything. So you guys, oops. Oh my God. I don't know what happened. Okay, are we good? All right. Woo. All right. Trends in Genetics 2020. Okay, so when many people think of eukaryotes, they picture plants, animals, and maybe some fungi, but these are just 3 very small lineages in the vast web that is eukaryotic life. So those 3 groups I just mentioned are all denoted by those arrows. So this is to say that most eukaryotes are protists, and protists are mostly microbial. These are some examples of what protists look like. So in 2019, a paper by the Bhattacharya lab and collaborators showed that the genomes of 13 Cyanidophyceae red algae were made up of 1% foreign genes. And this might not sound like a lot, but it is significant because these are small genomes that maybe only have about 6,000 genes, which would mean that upwards of 60 of them did not originate in the algae. So using this as inspiration, I looked for all of the papers that published a microbial eukaryote genome for which there was an accompanying analysis to quantify HGT. I found that the numbers ranged from 0.04% of the genome in budding yeasts to about 6.5% in a red alga. But keep in mind that this is only considering protein coding DNA, because at this point in time, there was a real focus on adaptive HGT or transferred genes that confer a fitness advantage to the recipient organism. Some examples of fitness advantages are HGTs encoding nutrient absorption, increased pathogenicity or ability to infect other organisms, drought resistance, energy storage, heat tolerance, and the list goes on. More specifically, here are examples of polar protists that have ice-binding proteins that have been traced back to prokaryote genomes. And these proteins allow organisms to adhere to ice and lower the freezing point so that they themselves do not freeze. And ice-binding HGTs have also been found in polar fungi and copepods that live under the sea ice. Copepods are animals. On the top here, I have some examples of where HGT was documented in some green algae and some stramenopile algae to encode salt tolerance traits that allow them to live in hypersaline environments. And then some diatoms like Pseudonychia and Fragiloropsis are able to sequester iron with ferritin, whose encoding gene was acquired from bacteria. And so this allows them to bloom in iron-limited waters, which is a trait that's absent from most other diatoms. And then an ulva, which is a multicellular green alga that we know as sea lettuce. You might find it on the beach. There's an HGT that encodes an adaptation to light fluctuations like it might experience in tide pools, and that gene is actually duplicated over 36 times. So clearly it's adding something important to the physiology of this organism. Many times throughout evolutionary history, organisms have made the lifestyle transition from free living to parasitic, and sometimes they have help from HGTs for that. So the oomycetes shown here are protists that live as plant pathogens, and they have HGTs from fungi that allow them to break through plant cell walls and utilize the plant's nutrients. And these genes actually make the oomycetes so convergently similar to fungi that when they were first discovered, they were misclassified for a while. HGTs have also been found that confer virulence or increased virulence in pathogenic ciliates and then in blastocystis, which is a gastrointestinal parasite. Last, HGTs have been implicated in facilitating some major evolutionary transitions. So I have a photo here I took of some choanoflagellates, which are protists that share a relatively recent common ancestor with metazoans or the animal lineage. They have HGTs that help them to be better predators and that may have enhanced their ability to become colonial and evolve multicellularity. In charophyte algae, which are the green algal sister to land plants, HGT from soil bacteria aided in adaptation to drier environments, and that may have helped early plants with their colonization of land. And then in Pollinella chromatophora, which is a photosynthetic amoeba with a recent primary plastid, HGTs compensate for functions that the photosynthetic organelle lost while undergoing genome reduction during organellogenesis. So these are just a few examples of how HGTs have impacted diverse eukaryotic lineages and aided in their survival over time. And there are more if you read the paper, which is very long. Okay, chapter 2. So chapter 2 is called Diverse Fates of Ancient HGTs and Extremophilic Red Algae. And somehow this is the old version of the PowerPoint. The new one had a really nice NASA logo here because this was one of the first projects I worked on as part of my NASA Finessed grant. So just imagine that it's there. So as my first chapter showed, it's pretty hard to argue at this point that eukaryotic HGT isn't a common and continuous process, and it does often lead to acquisition of useful adaptive functions. That was a weird noise. That being said, that's kind of where this work has stopped, and we don't know much about the principles governing HGT retention or integration into the eukaryote. Oh, I think somebody on this PowerPoint or on this Zoom is unmuted. But I can't see, so just mute yourselves. Okay. Anyway, so how can we study this? First, we need to pick a system. So the Cynidiophysce, as I mentioned, are a class of red algae that are found in geothermal habitats around the world. They've become great model organisms for studying HGT because they have highly reduced genomes. But like I said earlier, 1% of their genes are ancient transfers from prokaryotes. These genes encode functions like heavy metal detoxification, carbon and amino acid metabolism, and salt tolerance. And for this project, I just focused on the heavy metal detox HGTs. So I aimed to validate the function of arsenic and mercury detoxifying HGTs that were previously identified. We know that these sequences are in the algal genomes, but they were acquired a really long time ago. So it's important for us to understand what they're doing now. Second, I aimed to model patterns of HGT integration into algal metabolic networks. And third, I wanted to understand why these genes were able to stick around in these genomes for hundreds of millions of years and not be lost. So this slide, this slide does much better with the pointer, but I don't know. All right. So using two divergent Galderia species that split from a common ancestor about 500 million years ago, we're going to call them SAG21 and 5572. I tested my first aim to measure the gene expression of known HGTs with detoxification functions. And these include MER-A, which encodes a mercuric reductase that takes mercury 2 and reduces it to less toxic elemental mercury. We have ARS-A and ARS-B, which together encode the arsenite efflux transport system that pumps toxic arsenite out of the cell. ARS-C is an arsenate reductase, and then ARS-H and ARS-M together through a series of steps actually convert arsenite to less toxic forms. So the first thing I did for this project was run growth experiments to figure out how much arsenite and mercury would stress out our Galderia cells and induce a transcriptomic response, but not actually kill or really harm them. So I wound up choosing 5 millimolar sodium arsenite and 3 micromolar mercuric chloride for both species. I then set up RNA-seq experiments where I grew each species in each treatment and sampled at 4 time points across 1 week. And I extracted the RNA and sent from those samples, and I sent them to the Joint Genome Institute for sequencing. And this work was part of a large community sequencing project collaboration, and these are just some of our partners here. I analyzed the gene expression data through both differential and co-expression analyses. So differential expression is the classic way to see which genes are significantly up or down regulated under experimental conditions compared to the controls. And then WGCNA is a statistical method that clusters genes that are co-expressed across the entire experiment. So here you can see that mercury stress causes the MER-A gene to be highly upregulated at the 1 hour time point, but then expression declines back to control levels at the later time points. So this tells us, and this was in both genomes, so this tells us that the algae seem to have detoxified the mercury quickly and efficiently. Interestingly, though, the MER-A genes were not included in our co-expression analysis because they did not have high enough connectivity to any other genes. And this shows that mercury detoxification as a process is the 1-HTT, 1-function pathway, and it likely has not been integrated into the broader algal metabolic networks. Arsenic is a whole other story. So there are many more arsenic-related HTTs in these genomes. And as you could see, if you were to look at these heat maps for like 10 minutes, most are, or some of them are differentially expressed, but most are actually constitutively expressed, meaning that they're expressed all the time in both the control and arsenic stressed groups. One exception, though, are the 5 ARS-H paralogs in the SAG21 genome that all have a distinct pattern of differential upregulation with a peak at time point 3, which is 3 days. So I then looked at the WGCNA results with help from Erin Chili, who's an expert at this, and we linked the expression patterns of most of the arsenic HTTs with native algal genes and showed that most modules, which are clusters of co-expressed genes, share a common functional theme, and that they all had the HTTs all had high connectivity to other genes in the genome. The most exciting finding here, again, was also in ARS-H. So this gene in the 5572 isolate was linked almost exclusively with dark genes of unknown function and 1 photosynthesis gene. The orthologous, or the same gene in the SAG21 genome, was connected mainly to photosynthesis genes. And so ARS-H, the protein, is an NADPH-dependent FMN oxidoreductase. And photosynthesis involves the chemical transformation of NADPH. So this connection to photosynthesis started having me thinking that maybe the ARS-H gene is linking up with some other functions. The other 4 ARS-H copies were all in the same module, and they were tightly linked with translation-associated processes. One more thing to note is that while all 5 of these ARS-H copies shares a very similar expression pattern, the expression levels differ. So 2 copies have low expression, whereas the other 3 are highly expressed. And when I built a phylogeny for ARS-H, those 2 low expression genes also have very long branch lengths. And this further indicates neofunctionalization, or that they are evolving a new function. This is supported further by this multiple sequence alignment that I built to look at the actual structure of the ARS-H protein. So this protein has 2 active sites, and here you can see that the 2 genes with those long branch lengths and low expression values have an altered consensus sequence for NADP plus binding, shown in green, and then in one of them has an altered phosphate binding site to FMN, which is shown in red. So what does this all mean? I need a sip for this. So based on the findings of these lab-based experiments, we've come up with the integrated HGT model for eukaryote genome evolution, or the IHM hypothesis. So here we have an example of a hypothetical gene transfer from a prokaryote to a eukaryote following the introduction of an acute stressor, shown here by this brown gradient. Like with the mercury in my experiments, this requires a single gene response. The gene will be selected for as long as the stressor persists, but it does not integrate into host metabolic networks. And if the stressor diminishes over time, this lack of connectivity is going to prevent the gene's preservation, and an accumulation of mutations may lead to it becoming a pseudogene or non-functional. If the stressor was then to return, this eukaryote is in trouble and may go extinct, or alternatively, it could regain this HTT once again. And not only does this explain the low connectivity we see in these MER-A genes, but it may also explain why MER-A has actually been transferred to the Cynidiophysce lineage twice. The second scenario is consistent with the IHM. So here we have another hypothetical HTT, but this time the stressor shown in black requires a multi-gene response, kind of like arsenic. This stressor can be dealt with by one organism possessing an operon or by splitting up these operon genes among different organisms, and that would provide some redundancy and preserve the gene product functions of the pathway. So over time, the community response to the stressor will reduce selective constraints on these stress response HTTs, giving them space to evolve via duplication and divergence, and that's exactly what we see with ARS-H. So these reduced selective pressures also allow connectivity to increase. The HTTs are then going to integrate into various host metabolic networks. And then if this stressor changes over time, having these HTTs connected to diverse protein networks is ultimately going to prevent their loss. So this whole thing is just a hypothesis based on what we saw with MER-A and ARS-H and some of the other arsenic genes, but we did check to see if it holds in nature. So Tim Stevens analyzed metagenomes and metatranscriptomes from Yellowstone, and his work on the community-wide gene expression over the day-night cycle shows that the gene expression of Cynidioskizon and Galderia, which are the two different Cynidiophysce genera in Yellowstone, have an inverse relationship in nature. So Cynidioskizon is most active during the day as the keystone phototroph, and Galderia, which can live exclusively as a heterotroph, is most active at night. Mute yourself. Tim also looked at the expression of the same genes that I analyzed and showed that these genes are distributed among many organisms in all domains of life. And he concluded that Galderia, shown here in orange, is the main expressor of ARS-H, whereas Cynidioskizon, shown in red, is the main expressor of ARS-M. So two different parts of the arsenite detox pathway are done by two different organisms at different times of day. So if we combine my experimental results, Tim's Yellowstone results, and the ideas of the IHM, we can now understand more about how arsenic detoxification occurs in these algae. So I hypothesized that Galderia is resisting arsenite toxicity by pumping it out cyclically. And then once it realizes it's going to keep being exposed, it switches over to the detoxification pathway. And that's why we see that ARS-H expression peak at the 3-day time point. In nature, I also hypothesized that Galderia is resisting arsenite via extrusion, but then another co-occurring microbe like Cynidioskizon takes in that arsenite, converts it to a more toxic intermediate, and then Galderia ultimately detoxifies it. So those are the two hypotheses, and they also highlight why sometimes lab experiments don't tell us the full story. So it was really good that we looked at both lab and nature to understand what was going on here. So to conclude, we've used arsenic and mercury detoxification to compare two different HTT fates, and we've produced the IHM hypothesis. The complex arsenic gene expression in nature and culture indicates that there's a community level response going on with different taxa taking on different complementary functions. And this could support the idea of the earlier Earth as a cooperative community where both prokaryotes and eukaryotes are contributing to ecosystem maintenance. And we were really excited about these results, so we have future experiments planned where we're going to do some loss of function studies with ARS-H, and we're going to express it in E. coli vectors. Okay, halfway done. Okay, chapter 3 is called a K-mer-based approach for phylogenetic classification of taxa and environmental genomic data, and I swear in the real version of this PowerPoint that K is italicized. This paper was published last year in Systematic Biology. So as humans, we love to categorize things. In the 1800s, Darwin and Haeckel drew trees to depict relationships between organisms, but those trees were built largely based on morphological features. In the 1960s, Cavalli and Edwards began applying statistical approaches, including maximum likelihood, to build phylogenies based on blood group gene frequencies in humans. In 1977, Frederick Sanger created Sanger sequencing, and that allowed us to have access to DNA sequences to use in our tree building. And then in 1982, Penny, Folds, and Hendy then applied statistical theory to these sequences. So this extremely abridged history is all just to make the point that over time, advances in technology and computational capabilities have led to different ways to show hypotheses about evolutionary history. And there's really no reason why our progress should slow down now. So now we're in this era of fast and affordable whole genome sequencing, and we have so much data at our disposal that it seems wasteful not to use as much of it as possible. So why not use a holistic feature of the genome itself as the phylogenetic signal rather than marker sequences? And if we do this, our tree is not going to be built from a sequence alignment, and thus the method is considered alignment-free, or AF. So AF approaches bypass many issues that come from sequence alignments, such as assuming full-length contiguity of homologous sequences. They're computationally intensive, and those methods may present some issues that arise from inconsistent substitution rates or inheritance patterns within different parts of a genome or a cell. So the pipeline I used for this project was developed by the Reagan and Chan labs at the University of Queensland in Australia, and they used K-mers to reanalyze dinoflagellate phylogenies. So what am I talking about, and what is a K-mer? Well, K-mers are substrings of length K contained within a sequence. They could be words composed of letters within something like a book, or they could be nucleotides of a certain length within a longer DNA sequence. And so here I've shown an example of how the word Debashish can be broken into K-mers of two different lengths. So we have Debashish as 4 mers, or K-mers of length 4, versus 3 mers. All of the K-mers that make up that sequence represent that sequence's K-mer profile, and comparisons of profiles can be used as the input for phylogenetic tree building. Now, if we pretend that each of these boxes is a genome, the sequence that we're using to determine phylogenetic relationships is in red, and it's intact in this black genome. The green genome below it has none of the same words, and thus it would not have a matching K-mer profile. But because some of the words start with the same, or they have the same letters, and those letters are more or less in the same position, it would show similarity in a sequence alignment, and from that we would infer homology or shared ancestry. The third genome in blue would not align well against these other two because we don't see full-length contiguity on the part of these red sequences. But if we broke, but if you look at the red words, they're actual words, they're the same, they're identical to the words in the black genome. So if we broke all 3 genomes down into K-mers and compared the K-mer signals across those 3 genomes, now we would find that the black and the blue genomes are more closely related. I think I forgot to mention that in the first example, the black and the green one were more closely related. Anyway, this illustrates how 2 different methods would lead us to 2 very different phylogenetic inferences. So for this chapter, I asked if these AF methods look at such a large amount of data, can they be agnostic to some of the missing parts? So here I first aimed to compare AF K-mer-based methods to traditional methods using well-known algal taxa. Second, I wanted to simulate lower quality fragmented genome data using these algae to test our method's robustness to genome quality and completeness. And third, I wanted to apply the AF approach to some real data sets from environmental or single cell sequencing projects. So the first thing I did was I chose 3 well-studied algal groups to work with. I picked the Cyanidophyceae, which are red algae, the Chlorella-clade, which are green algae, and then the Nanochloropsis genus, which are Acrophyte, Stromatopyle, or Heterocont, or whatever you want to call them. I think people are arguing about it right now. I took these photos of each of them, and as you can see, they all appear as little green dots. So this morphological similarity masks hundreds of millions of years of divergence. And that's exactly what a bunch of people hundreds of years ago saw in their microscopes when they were first characterizing these organisms based on how they looked. And they were historically misclassified as one another. But we now know from molecular data that they're not even very closely related. So I thought they were 3 good groups to choose for this project. I ran a pipeline on these groups of genomes that we adapted from the dinoflagellate projects. So in short, we create streamlined versions of the genomes by masking repetitive regions. We choose a value of K to use. Then we build a distance matrix of pairwise K-mer frequencies between all of the genomes in our set. We use that matrix as input for building our phylogenetic inference, which in this case is a neighbor joining tree. And in contrast, I also built the same phylogenies using a traditional marker gene-based approach where I built multi-gene maximum likelihood or ML trees. I compared each set of AF and ML trees visually using tanglegrams, and I also generated normalized Robinson folds distances for each tree comparison. And the RF distance is just a metric of how many changes to branching occur between the two trees. So all of the trees here, the takeaway is that all of the trees here are very similar, and that shows us that this AF method is a valid alternative for reconstructing phylogenetic relationships within these algal groups. To address the second aim, I used an approach similar to jackknifing to randomly remove percentages of each genome in chunks that range from 1,000 to 50,000 base pairs. So each percentage chunk combination was replicated 50 times to produce 2,700 simulations. The altered genomes produced by each simulation were each used to generate their own distance matrices and neighbor joining trees. And then for each one, an RF value between it and the reference K-mer tree from AIM1 was computed. So 2,700 trees and RF values. So this was done 3 times. So for all 3 algal groups, and the RF values were plotted as density points. These are just the chlorella results right here. And the takeaway was that most of the time, until you get to the most highly fragmented, so say 90% of the genome is deleted by the largest chunk size, so say 50,000 base pair, until you get to that point, the trees were more or less the same as the original. And I even ran subsequent analysis using PhySort-R, which is a program that Tim created. And that showed us that the major clades and basal topologies were all conserved throughout. And it was just on those terminal branches that things were switched around, even in the trees with the highest RF values. So the same trend held true for the other 2 algal groups. And that shows us that the AF method we use is robust to some pretty extreme genome degradation. For the third aim, I chose 3 real data sets of varying quality, and I included a prokaryote example and in the Saccharomyces bacteria, and then 2 eukaryote examples. So I picked trabuxiaphyte green algae and then Stramenopile single cell amplified genomes or SAGs that were from the Terra Oceans project. And the results of these 3 analyses are going to highlight the limitations of traditional methods compared to the AF methods. So the Saccharomyces bacteria, I think they're really cool. They were a newly discovered phylum. So they're part of this candidate phyloradiation that we didn't know existed until recently. They've only ever been identified from environmental genomic data. And to my knowledge, I don't think they've been grown in the lab. So we found 12 novel Saccharomyces metagenome assembled genomes or MAGs in Yellowstone. So I used those along with hundreds of others from projects around the world to build these trees. Like with our model algal data sets, the AF and ML phylogenies compared pretty well, which shows us that the AF method is a valid alternative. However, I would actually argue that this AF method is much better because the available numbers of marker genes for each MAG were extremely inconsistent because these bacteria have really reduced genomes. And in the literature, there's also a lot of talk about how the Saccharomyces have highly divergent and like really funky 16S sequences. So that kind of renders traditional maximum likelihood gene tree building unreliable for these bacteria. Then 2 of our Yellowstone MAGs were also preliminarily classified as unidentified trabuxiaphytes. So I added these to the chlorella clade tree from the first name because chlorella are also trabuxiaphytes. So here for the ML tree, I used single copy Busco orthologs to build the alignment and build the tree. However, there was no single Busco gene found in all of the genomes. So I used a subset of 27 genes shared by 90% of the genomes, which means that the alignment I used to build the ML tree contained a lot of fairly large gaps. So these trees compared okay, and mostly just the terminal branches were affected while the basal topology was similar. And then last, I compared the Stramenopile AF tree to an ML tree that I did not build. It was built by C. Luthner et al. 2018 as part of the Terra Oceans project, and they used 160 proteins for their alignment. So the same issue here arose where some of the SAGs or the genomes in this tree only had a few dozen of those proteins and others had over 100. So their alignment had a lot of really big gaps, and that is evidenced in their tree by these really low bootstrap support values at the most basal divergences. So again, I suspect that the AF tree that I built is probably much better and more reliable. However, there's really no way of knowing without knowing the precise evolutionary history of these organisms. And so this highlights why tree building is such an imprecise endeavor to begin with. So in the future, AF methods can help standardize this subjective type of analysis that's highly biased by user choice. And like Lynch and Neufeld said in their 2015 paper about the rare biosphere that I really like, uncultured and rare organisms likely account for a majority of the species on Earth. So you might find a partial genome sequence if you're out in the environment sequencing DNA. You might find a partial genome sequence that has no marker genes, and it might be from some sort of novel organism you're never going to find again. So an AF method like this might be your only way to explore this unknown biodiversity. And I think this rings particularly true for rare protists. And through this project, I've shown that these methods work on mags from 2 different domains of life. And I also think they show promise for museum, herbarium, and ancient DNA samples going forward. Okay, last chapter. This one also had a really nice, pretty NASA logo on it that didn't make it into this final version that somehow they uploaded on here today. Anyway, this chapter is called Genetic Transfer in Action. So similar to the work of chapter 3, for this project, I use another alignment-free tool that was made by our collaborators. And in this case, it's based on a statistic called term frequency inverse document frequency, or TF-IDF. And I'm sorry to give you another acronym. This statistic was originally used in the field of documents analysis to find words that are rare in one document, but frequent in a broader collection of documents within a database. So it teaches us about like word significance. Yingnan Kong and Mark Reagan adapted this in 2016 to look for HTTs in bacteria and archaea genomes. But upon reading about how it works, I thought it was really cool, and I wanted to see if we could apply it to eukaryotes and to lower quality environmental genome assemblies. So here, I aim to use an alignment-free Kamer-based method to uncover coding and non-coding DNA transfers using environmental genomic data. And I wanted to create a filtering pipeline to validate the results of this method, because the program might run, but how do we know that the results are any good? So after filtering, I wanted to use those results. Third, to infer ecological relationships and putative mechanisms of transfer from within this single microbial community. To illustrate how this particular Kamer method works, here we have a brown genome that's very different from the blue genome beneath it. We can assume the brown one exists as one of many closely related genomes in a database, and so does the blue one. Using the TF-IDF statistic, we would identify words or Kamers that have a low frequency in the brown genome, but a higher frequency in the blue and blue-related genomes in our database. We would then extract those Kamers from the genomes and inspect further to see if we think a DNA transfer occurred. So for example, if we broke down our brown sequence into 4 mers, or Kamers of length 4, we'd see a lot of LOVE strings, or the word love, and then we'd see 1 hate here in red. And then if we did the same for the blue genomes, we'd see a lot more hates. So using that information, we could then infer that there was a transfer from the blue to the brown genome of the untrue phrase, Debashish hates rhodophytes. Okay, so for this project, we again used our Yellowstone metagenome data set. So these samples were bravely collected by our collaborator, Tim McDermott, during the lockdown from various habitats within a site called Lemonade Creek. So I did the lab work here to extract the DNA and RNA from these samples, which were then sequenced by the GGI. Tim Stevens did the binning and assembly of the algal and prokaryote mags, and then Felipe Benitez built the viral assemblies. I then ran a modified version of the TF-IDF program and created a pipeline to filter the results to a conservative subset that were validated and could be individually assessed. So in short, there were over 13,000 raw results, but after applying size filters, discarding misassembled scaffolds, simple repeat sequences, results with coverages that deviated from the rest of the assembly, et cetera, et cetera, et cetera, we ended up with we ended up with 39 exactly robust HGT candidates. Most of these results were within domain transfers, so they represented putative HGTs that were from bacteria to bacteria or archaea to archaea. But of the 39, over half of them were non-coding, and that was supposed to be all in bright yellow because it's really important. And that tells us that HGT shouldn't only be evaluated on a gene basis. Of the 17 that were potentially protein encoding, 10 had some homology to annotated proteins, and they included two transposases, a restriction endonuclease, a DNA methyltransferase, and an S-layer protein. I also built phylogenies for all of the protein coding results, and I found that many of them do appear to be bona fide HGTs, meaning that the transferred sequence is closer to other sequences, or it's more similar to other sequences of the donor or relatives of the donor, rather than sequences related to the recipient class. And this is just one example of a nice looking tree from this analysis. Then I built a network of the 39 transfers to show the breadth of this process across the whole community. So with respect to the algae, there were associations with Saccharomyces bacteria, Pandora viruses, and a potential fungal transfer. Then we have our transposases. One was transferred between archaea, one was transferred between bacteria. And then between the Thermoprotea and Thermoplasmata archaea classes, there was evidence of a restriction endonuclease and a DNA methyltransferase, which is really cool because those two genes together make up the restriction modification system. So these results provide a basis for forming hypotheses about mechanisms of DNA transfer and ecological relationships. Mobile genetic elements like transposons that encode transposases are known facilitators of DNA transfer because they enable DNA to jump from one place to another. The restriction modification system components that were found in the archaea, they make sense as a way to mediate which transferred DNA gets to stay in the new genome, and that helps us understand more about how HGT gets done. And all of the results taken together, so these and a lot of other ones I didn't have time to discuss, they make me confident that this method worked well for uncovering those gene-independent DNA transfers. Also, by looking at the algal results specifically, we can start to form some loose hypotheses about maybe a possible symbiosis between Gaulderia and Saccharomyces, which are known symbionts, and then possibly a host pathogen relationship with the Pandora viruses. And perhaps the viruses are mediators of algal DNA transfers, but that's something that we'd have to investigate specifically in the future. So, overall, this project is a good first step into looking at HGT as a community-wide phenomenon that's driven by ecology and drives ecology. However, this work will need to be replicated in other diverse environments. Future work should use long-read sequencing data rather than short-read, which we used here and it was stressful sometimes. Ultimately, this kind of analysis can help us understand the background levels of HGT across our entire biosphere. So today, we started out with HGT and eukaryotes, not if, but how much. And now I feel like I'm leaving you at, well, there's a lot of HGT in all domains of life, but how, where, and why does it happen? So my work through the latter 3 chapters here answered some of these important questions, but I am excited to continue working on this and to see where this whole field goes in the future, considering how much biodiversity there is on the planet that still needs to be explored. Thank you. That's it.
Did a great job.
I have so many. I have to do a thank you. Okay, I have like 5 minutes of thank yous to do. Okay, so I want to thank the Ecology and Evolution graduate program. I want to thank the Department of Biochem and Microbiology. I want to specifically thank Shanika, Jesse, Marcia, and Mike Pierce. I want to thank NASA for funding most of my PhD. In the new version of the PowerPoint, I had SGS and the Bevier Fellowship, which funded me, which was great. I want to thank the DEI committee and EcoGSA for their moral support. I want to thank the JGI for funding all of our sequencing. I want to thank our collaborators and co-authors, especially Tim, Hwansu, Colin, and CX Chan in his lab. I want to thank the Bhattacharya lab members, past and present, especially Tim, Aaron, Alex, and Arwa, who helped on all of these projects. And I want to thank the best committee ever, Lena, Siobain, and Lynn, who I really could not have picked a better committee. You guys are the best role models. It was no accident that I wanted 3 really brilliant female scientists on my committee, and I got that, and it's been great. And then last but not least, I want to thank Debashish, because I can't think of a place that I could have been for 5.5 years and learned more. Like, you're a great advisor and a great mentor, and I've had a great time doing my PhD in your lab, and I've learned so much. Here are some photos. Here are some photos of some fun Bhattacharya lab times, lots of stuff in the field or at conferences or Halloween or at your house. And then, okay, so then I want to thank my sister Nicole, my dad Justine, John, my uncle Art, Murray, Ella, Phoebe, all of my family, all of my friends, especially a bunch of you here, Emma, Dylan, Erin, Gabby, Kathleen, everyone. And then last, I want to thank my mom, because I wouldn't have been able to start or finish this program without her, and she's helped me so much. Thank you.
I believe the word you use on the slide is best advisor ever, not great.
Well, I'm still trying to pass the private defense later, so.
Smart move. All right, so that was the public lecture. Now we're open to questions from the public, and
There's so much in the chat.
let's start with the human beings in front of us.
Okay.
And we can move on to, are there any questions from anybody?
This relates to the theory and fact too. Is there evidence that mercury is possibly exposure was more sporadic throughout the evolutionary history?
So that's a really good question, and I have looked into this, and it is so hard to know. I think the answer is probably yes, but these geothermal environments, like the turnover is kind of high. So like they're like the ones we're looking at now are kind of new. Like, they're not billions of years old. So I don't actually know, but I looked in the literature, but I'm not sure. Peter.
I was trained originally as a plant breeder. I'm watching this and thinking, maybe I should just get out of the way. And that's all plant wide or larger, not that we find it successfully on this place. And watch. Are we just wasting our time, or are we actually moving things along in a coherent way that is somewhat compatible with what would happen anyway?
I'm not sure I understand the exact question. Like, can you ask it again?
Well, it's gonna happen, whatever it is,
Okay.
okay? Doesn't need us.
Just like evolution and
Evolution on the planet,
sure.
okay? So why are we wasting our time?
Because we're curious. No, well, there's, I mean, so something we talk about a lot is like there's an ongoing biodiversity crisis. And I personally believe that one of the ways to help with that and to help humanity is to try to understand what organisms are even out there. Like, I think a majority of microbes are unknown. So if we don't even know what's on the planet, how could we even start to think about what to do about our problems? So that's something I think about a lot. I don't know if that answered your question.
It's close. Okay.
Okay.
Let's go with John. Go first.
John, you have a question? Okay. So like in eukaryotes specifically, it's like really hard to know. Like, what I think is that it's happening, it's a continuous process and it's happening all the time, but we're only seeing the transfers that were selected for by natural selection because they provided some sort of adaptive advantage. But my 4th chapter showed that there's a lot of like non-coding stuff that gets transferred that probably gets purged from genomes really quickly. But the ones in the 2nd chapter with the heavy metals, those are really ancient and they persisted because they integrated well in these genomes, and they're probably still necessary. Yes.
Joe?
Number of my favorite fungi are good at metabolizing arsenic. How can I find out if those genes are
Oh, yeah. So the classic way to look for an HTT would be to pull out those genes from the fungi and to build a tree with a lot of diverse genes from across many taxa, so bacteria, archaea, etc. And you're going to want to see if your gene tree is different from the species tree. So if that gene has a different evolutionary history than the fungal species you're looking at, that would be how I would do it.
Other questions?
Yeah, awesome.
Do you have a sense, is there a way of dating these events of lateral gene transfers that you described?
That's a really good question, and that's something I'm thinking
Can
about.
you repeat the
right now.
question?
Ooh, 1 second. Oh, repeat the question. Oh, he wanted to see if there's a way to date these transfers. I'm assuming from the 4th chapter. So this is something I'm thinking about, and it's hard to, there are probably some like molecular techniques I don't know about yet to date them specifically, but I do think that there are ways to relatively date them by like, part of the filtering I did was looking at a lot of different features of the sequences. So like GC content, and things like that. So I think there's ways to relatively date them. Like over time, these genes ameliorate and they like look a lot like the genome they've just joined. So looking at differences, like looking if they still look like donors versus if they look like the recipient would be a way to relatively date them. Lena? Okay.
So if you have a phylogeny of, say, lots of algae, and you can say that there are many of them that have this HET gene in them, then you can date it down that it happened at least.
Oh, sure. Yes. Yeah. I was thinking more about like the gene-free ones, but yes, you could totally do that. So if you build a tree and a lot of them have it, you can pinpoint like if it was donated to an ancestor versus if it like went to a specific single lineage, which is how we know about a lot of them in the Cynodiophyceae. A lot of them were really ancient transfers, maybe 1 billion years ago to the ancestor of all Cynodiophyceae. Did somebody in the chat or in the Zoom have a question? I can't really see. Like I can't see you, but I can hear you.
They can unmute at that point.
Oh, okay. Yeah, someone can, if anybody in the Zoom has a question, you can unmute. I just can't see anyone because this screen I'm on is really confusing. Maybe if I stop the share. Oh, now I can see people. So if anyone in the chat has, maybe there's questions in the chat. I can't tell. No, I don't think so. Nice messages from everyone though. Thank you. Anything else?
Other questions from the audience? If not, then thank you all for coming and thank you, Julia. All right, so we will take about a 15-minute break and the committee will
Oh my
convene,
gosh.
Julia and Ferran, all 262. And I'll have a little bit of food there. If you're hungry, you can snack on it.
Oh my God.